welcome to RCA Radio, a podcast where we cover the latest news and challenges in regulatory, compliance, and quality assurance facing the life science industries. I'm your host, Brandon Miller. In this episode of RCA Radio, we'll be taking a dive in the world of preclinical testing for medical devices and biocompatibility for launching your products. Today, I'm joined by Dr. Matthew Jorgensen and Dr. Helin Ragel. Matt is a board-certified toxicologist with a PhD in chemistry and is Nelson Laboratories chemistry and materials scientist. And Helin is Nelson Labs senior biocompatibility expert with a PhD in cell biology. Welcome, Matt and Helin.
Yeah, we're happy to be here.
Yeah.
Thank you both for joining me today. I'm very excited for this. Today, I want to pick both of your brains to help educate our listeners about the world of preclinical testing and biocompatibility for medical devices. And I want to just cover some of the basic background of preclinical testing, explore some new things happening in the field today, go over some best practices for testing, and then end on some advice for picking a testing lab for yourselves. So let's just jump right into it. Matt, Helin, can you guys give me an overview of exactly what preclinical testing and biocompatibility are? How does it fit into the entire product life cycle, and why is it important?
Yeah, absolutely. And we are so happy to be here and talk about this. It might be kind of hard to believe, but biocompatibility and ensuring that the devices that we help make it to market, to ensuring that those are safe is really a passion for both of us. So it's lovely to be here and share some of that. As I was preparing for this podcast, I went to the library and I checked out a book on abdominal pain, but the appendix was just ripped out. so there's a couple of holes missing. And when I think about ripping stuff out, it kind of like leads me into thinking about medicine, the devices that we use. And when I start to go down this path, looking at biocompatibility and why is it important, I always remember Hippocrates, the Greek physician Hippocrates. He's attributed with the formulating the Hippocratic Oath, of course, which is central to modern medicine. And embedded within that is this idea, first, do no harm. Now, that statement, first, do no harm, is technically not in the Hippocratic Oath. There's other statements that mean the same thing. But this idea that within the field of medicine, both in pharmaceuticals and clinical practice and with medical devices, we want to ensure that whatever we're doing when we intervene with patients, when we try to treat them and make them better, that first, we're not doing anything that might harm them. And unfortunately, there's lots of examples where well-meaning people, doctors, medical device manufacturers, in their attempt to actually help people lead better lives and thrive, have ended up accidentally harming them. So there's lots of ways that treatments can go wrong. One of the most famous examples is a hip replacement. Sulzer Orthopedic is now a classic case study. So they manufactured a ball and hip replacement, and they manufactured these. They made some manufacturing changes to bring part of the process in-house. I don't want to get too technical, but after making some changes to their manufacturing processes, patients started to complain. Patients were complaining of burning and really like hot pain where the hip replacement was. Doctors started to investigate. There was like hips sort of like slipping out of the socket. And this situation escalated. People were really, really uncomfortable. And once they started to go in and look, the bone wasn't integrating with the hip cup. And they were forced to make a recall. And this was after 17,000 of these hips had already been implanted into people. The financial impact of this onto the company was astronomical. So at the time, it was $1 billion. That was the value of the settlement to start to make this right. But if you think about the impact this had on patients, they went to their doctor because they were having trouble walking. They needed a new hip. They got a new hip, but actually this ended up harming them more than it helped them. And the only recourse for most of these folks was to have revision surgery. So they had to attempt to remove the old hip and put a new hip in. But in lots of those cases, a revision history wasn't possible at all. So these folks could never walk again. So the impact when a medical device goes wrong, when it's not biocompatible, can have far-reaching consequences that all of us want to avoid. So within the regulatory and medical device business and within the field of medicine in general, obviously we want to first do no harm. We're here to help people lead better lives. We want them to thrive. And making sure that medical devices are biocompatible is central. It's key to that. There's other examples of how, that have raised awareness of why biocompatibility is so important. If you want to, have a good cry, just Google thalidomide and take a look at the photos on Google Pictures. In a nutshell, thalidomide was a pharmaceutical that was brought to market in Germany in the 1950s. They had done some testing on its safety, but not in pregnant women, and they had thousands of babies born with birth defects because that developmental toxicity wasn't one of the things that they were really checking when they were bringing evaluating the safety of that product. So they were trying to make people better. It was marketed as a sedative, and it ended up doing almost unimaginable harm. So I just mentioned two ways. One way that a device might not be biocompatible is like with the case of the implant, where it doesn't have good tissue integration and that causes problems. Another is a more systemic toxicological problem that led to developmental toxicity concerns. Over time, the community of people working in medical devices have figured out lots of different ways that devices can harm people. And this has been consolidated together into the most important ones that need to be checked for. And for that, I'm going to turn it over to Dr. Ragel, who will discuss the main ways that a medical device might inadvertently harm somebody, and then also the tests that we do to check and make sure that those aren't an actual concern.
Yeah, so like Matt was telling us that it's really important to make sure that the patients are not faced with some adverse reactions while the medical device is being used on that. Or maybe even like in case of the hip implant, it's like permanently incorporated into their body, right? And over the years, we've gone a long way since Hippocrates. So we have noticed that there are specific endpoints, that's what we call them in the biocompatibility world, specific biological endpoints that we really need to assess for when we are taking a look at medical devices. And these endpoints are really dependent on the duration of contact and the type of contact that the medical device will have to the patient, right? Obviously, something that is put on in contact with skin, right? That would have much less impact in terms of what should be checked for than a hip implant, or something that would maybe even go into the bloodstream, right? Because, bloodstream to me is like, oh my goodness, that's the worst of the worst case hazard to me, because if there's something going on, the response could be totally acute and, we don't want that to happen. So over the years, the regulatory bodies and industry, they have formed these standard documents, right? For example, ISO 10993, and specifically ISO 10993-1 is, I call it, the Bible for biocompatibility. So whenever you have a product that you want to take to the market, that is the first place to consult. Right. So you take up, the ISO document, either on paper or, the new way of doing things online. So you kind of take a look. There's a very, very good table in there that talks about the device contact duration and type of contact that the device will have to the patient. It's called table A1. That's like, I close my eyes, I still, see it. That's how much I've seen it. So then you kind of take a look at the endpoints, the biological endpoints that the whole industry and the regulatory bodies have together decided that these should be really honed in on when you look at your own medical device, right? So for example, when we have a medical device that contacts the skin, right? Then the specific biological endpoints that should be taken into consideration when you evaluate your medical device, and make sure that you realize I'm saying the word evaluate. So when you evaluate your skin contacting device, you need to look at cytotoxicity, irritation, and sensitization.
Hey,
right?
Dr. Ragel, can I just jump in here and say two things? First of all, you said we should start by referring to table A1 of 10993-1, but I would also, add an asterisk there that folks can always call us up. Our role within Nielsen Laboratories is as experts, and we are very happy to take folks and talk them through this process and help them pick these endpoints. So that's number one. Number two, you've used a lot of words just now that I don't think are common to everybody. Can you just say what cytosensitization and irritation are?
Absolutely. And of course, you can call me up. I would love to talk to you about biocompatibility. Let's talk about the big three that we call the big three is like the main endpoints, the cytotoxicity, irritation, and sensitization. Okay. So cytotoxicity is a test that we run to understand if we put your medical device into extraction solvent or like a liquid, right? What could be coming out of there and whether that extractables profile, that's how we call it, whatever is migrating out of the device, does that have potential to cause cellular destruction? Okay, so basically they will look at whether it kills the cells or not, or affects their morphology or viability somehow, right? So that's the first thing to check.
So basically will your device kill cells? That sounds pretty important.
I bet that hip implant maybe could have caused some cellular destruction. Okay, so it sounds pretty straightforward and simple, right? Cytotoxicity, we put it on cells, we see cells die. But actually, there are quite a lot of minute details to that testing. And because it's an in vitro test, most of the other tests are in vivo tests for like the irritation and sensitization. It does tend to be more sensitive. And with that comes the array of, if we do see some reactivity, is it actually going to be causing a harm? Again, hint, hint, if you have this issue, you can always call us. All right. So irritation is the second point, right? So basically we want to know if your device is going to contact the patient, either the skin or the tissue. We want to know if the extractables profile, again, what can be coming out of the device when it's, exposed to like a solvent, is that going to cause irritation in the tissue or skin? And what we do a skin sensitization test where we look at reddening or swelling of the site where we have the solution that has come off from your device being put on the skin or inside the skin. Sensitization is similar type of reaction where again we're looking at irritation or the reddening or swelling of the skin, but sensitization is a type of allergic reaction. It's kind of like poison ivy. When you have the first time exposure, you might not even do anything or feel anything. So you might think, I'm cool. I don't have a reaction to that at all. But then when you have a second exposure or third exposure, then you'll be like, I see this rash. It's kind of like Coraline with the little rod, having the poison oak in their hands and then having the reaction on the skin. Okay, so we don't want to, go down that path, right? So those are the main three, the so-called big three, that we need to evaluate for any medical device.
Okay, I'm just going to restate what you just said. I thought it was perfect and beautiful. So basically what we're saying is that for every medical device to go to market, it has to be evaluated. Does it kill cells? Is it fatal to cells? Will it give you an allergic reaction? Or if you put it on your skin, is it going to cause redness and swelling? It seems clear and straightforward. Those are things I don't want to happen to me when a medical device is used on me. So those are the big three. They're required for all of them. We don't want to kill our own cells. We don't want to give people an allergic reaction, and we don't want to cause redness and swelling. Okay,
yes.
Sorry. Continue.
Exactly. And then obviously, the more invasive your device is, additional endpoints would become relevant as well. For example, if you have something that is going to be inserted into your body, we need to make sure that no systemic reaction, like for example, acute toxicity is not going to come, that you're not going to have anaphylactic shock, right? From the extractables profile, that's, there could be something that will either impact the tissue or the blood. If you have a catheter, we need to also make sure that interaction with the blood is not going to cause harm to the patient, right? For example, clotting. Because if you have some catheter inside the system and there's blood going around the surface of the catheter, obviously you want to make sure that the leukocytes or the red blood cells that are coming into contact with that device are not going to be harmed in any way. And obviously, for every single one of these endpoints, there is a test. But remember, I did say evaluate, right? And with maybe we want to talk about like the evolving of the standards right now to go into the details of that.
Yeah, I do. I want to talk about how the standards have evolved and the methodology or thinking behind how do we make sure that these risks are fairly and adequately addressed. But before I do that, I thought of a case study, a certain device when you said acute systemic toxicity. So acute systemic toxicity, that checks for death in the near term. And it seems like that would be pretty rare to happen, but it has happened a couple of times. Mm-hmm. We had one device, it was an ablation catheter. So this is a catheter that goes into the body. It gets very, very hot to burn tissue during surgery. And we had one of these that we were testing, and we did an acute systemic toxicity test in vivo. So we used the device, extracted it, and exposed mice. And the mice were dying very, very fast. And so then we got, we're like, kind of like interested. Like, we see mice die slowly sometimes, mostly from being lonely, but but they were dying very, very fast. And so we started to look into this a little bit more, and it turned out that there was a legitimate acute systemic toxicity concern. The tip of this device, the ablation catheter, had ABS plastic. It was ABS that was produced not, for medical grade. It was actually from a supplier that mostly makes car bumpers. And it turns out that when you heat ABS, it can release cyanide. And actually it was that the mice were dying so fast that we could narrow in like, okay, there's only 2 or 3 poisons that are capable of killing a mouse that fast. And cyanide was one of them. And sure enough, when we checked, it was indeed cyanide. Yeah. So this is, so I'm just very, very grateful. It's almost a funny story, but on the other hand, I have parents and grandparents in my life. They've needed surgeries in the past. I'm so glad that these things are being checked for. It would be devastating to have this used on a human and have them die on the operating table due to cyanide. Mm-hmm. Okay, but now I'm going off down a tangent there. Helin was just saying table A1 of 10993-1. It lists all of these potential concerns that need to be evaluated when we're thinking about biocompatibility. The big 3 that she said: acute systemic toxicity, other subacute and subchronic toxicities, carcinogenicity, and so on and so forth. And the question was, or the lead up was, well, what's new? How has this been changing? I'm just going to talk about that really briefly as we lead up into the most modern way to evaluate biocompatibility. So historically, these endpoints were recorded and embodied in the FDA's manual G95. And this was viewed in a very checkbox, like linear way. So you had these set of biological risks. For every risk, there was a test. And folks that wanted to bring a medical device to market would simply order those tests on the list. If they all passed, great. The device was clear from the perspective of biocompatibility. The problem with that approach historically was, one, it meant that so long as you passed, that was the only thing that mattered. You could cobble together a device using stuff that you found in your garage and fishing line that you picked up from Field and Stream, true story. And it could pass, and that was it. You didn't even need to know what your device was made of, and it could pass biocompatibility. So that was a problem. The other problem was we had things like devices, let's say a screw made out of titanium 6Al4V. So that's a very, very common titanium alloy. And we've tested TI 6Al4V maybe 10,000 times, maybe even more. And we probably have TI 6Al4V samples on test right now downstairs. It doesn't need to be tested over and over again. It makes no sense. I don't want to put titanium screws in rabbits and show again that it's not an issue. So it meant that people were just testing regardless of if we already know that the thing is safe. So there was a critical transition there where we moved away from saying everything has to be tested to everything needs to be evaluated. So for all of those biological risks that are in 10993-1, table A1, we still need to address all of those. We need to know why this thing isn't going to cause a sensitization reaction. We also need to know that, this isn't going to cause your patient to die within 30 seconds of device use. Why is acute systemic toxicity not a concern in cancer, and so on? But now the window is really open, and I don't even want to say open. Now it's actively encouraged that you avoid animal tests to evaluate those endpoints. So that's a really critical pivot point. We're going from, just accepting animal tests or just routinely doing animal tests to only performing those when it's absolutely required, when there's evidence that suggests that those tests need to be done. Then we could go into the nitty-gritty details of how these standards have changed and what that means, how it's being incorporated. But that's really like the core message, both in the US and all through Europe, really the world. You should only test an animal if you have to. And you should use every other means available to evaluate those endpoints otherwise. So that means that a couple of other things have come into play. One of them is chemistry testing. As I was introduced, I'm a chemist. I love chemistry, almost more than anything else. So I'm really excited about that. Today, we do a lot of chemistry testing on medical devices and then follow that testing up with a toxicological risk assessment to address all of these systemic toxicities that could come into play. So chemistry testing is really big right now. There's a lot of detail in the expectations of how that chemistry testing should be conducted so that it's satisfactory to toxicologists and to regulators. I won't go into that here, but we could have another 3 podcasts on just chemistry testing alone. I don't know, Helin, is there anything else that you want to talk about with what's new with respect to the standards and the way things are moving right now?
Mm-hmm. Yeah, I do want to also plug that like the 10993-1 is now currently saying is that you need to have this upfront evaluation, like Matt was saying, that you need to take a look at what is your medical device, how it contacts the patient during use, and what do we know already about the materials and processing of it to make sure that we have a documented so-called a risk assessment, or what we call biological evaluation plan, that goes over all of this background information and available knowledge, and then defines what specific endpoints need to be followed up with additional testing. Right? Like, the titanium implants are a great example because we do see so many of them and it's so ubiquitously used as an implant material, right? So we know that the material itself is not really harmful. But we do need to also take into account the manufacturing process, right? And to evaluate that, there's different ways that biological evaluation plan should really address, right? Again, the point of the biological evaluation plan is to summarize all the available information and then define what additional testing should be conducted to show that indeed there are no unacceptable adverse effects happening during the exposure to the patient, right? So, and expert advisory services here at Nelson Labs, that's, one of our bread and butter. I do those fairly regularly. So again, call me up, call us up, we can help. Then, as I said, that should be the first step, right? To identify what should be tested. And then the second step is to actually perform those tests, right? And now we need to kind of cover like, okay, there are many different labs out there that could perform some of the testing, right? So how do you pick, Matt, how do you pick the ones that could give you quality data that is really needed to get you through the regulatory submission? How do you choose the right lab for your testing?
Well, I understand that there's lots of labs out there, and I think it's great that so many people are in the business of helping medical devices to be safer. I think if I put myself in a manufacturer's shoes and I want to select the lab that's best for me, one of the things that I would really want is a depth of expertise, both in the lab and in the advisory services that helps to support the lab. So for example, you mentioned cytotoxicity testing. It's a very sensitive test that commonly fails. I want to know that there's people there in the lab that can help in case of a cyto failure. So when something goes wrong, it's not just silence and a failed report, that we can have a discussion, that there's people there who have seen this before and can come in to aid. So I think about Audrey Turley. She's somebody that's here that has decades of experience with cytotoxicity testing. She's probably run more of these tests than any other person alive. Not only has she run them, she is an expert that informs ISO 10993-5. So she's helping to shape the future of that standard. And then we have you who talk to the FDA on a weekly basis or more and really understand the pulse of regulators when it comes to how this test should be applied and used. So as people look for labs and want to approach this testing, I would say start with expertise and also history and background, so that when there's an issue, if there's an issue, that you have this support framework around you that can get you through that. The other thing that's really important, as I mentioned, I'm a chemist. I love chemistry. I can't not talk about chemistry testing. Since we move away from animal tests and we supplement those or replace that evaluation with chemistry testing and a toxicological risk assessment, the way that the chemistry is executed is very, very important to regulators, especially to the FDA right now. So I would be anxious to know that the lab can test the chemistry in a way that's acceptable to regulators. There's a lot of detail there. Regulators really don't like unknowns popping up in their chemistry reports. If you have an unknown above a threshold of toxicological concern, that could be a deal breaker for some reviewers. And so you want to have a lab that's doing chemistry that can minimize the number of unknowns. The way that happens is if people have been doing extractables chemistry testing for a long time and have built up a very robust, very large library of compounds, we can really help to avoid some of those issues. And that's one of the things that we enjoy through Nielsen Labs in Leuven. They've been testing medical device and pharmaceutical materials for 30 years. And over those 30 years have just amassed compound after compound in this library that really helps to minimize the number of unknowns.
Mm-hmm. Yeah, and then that helps the toxicologist to actually determine what is coming off, how much is coming off, and then define whether those are within the acceptable limits for the duration of the device. So you could get chemistry data from, let's say any chemistry lab, but the usability of the data, that becomes the make or break points.
Okay. So I think to edge towards closing out, if you could, in 2 or 3 sentences, just sum up the entire biocompatibility process for a medical device, what would that look like beginning to end?
Okay. Yes. So because that is the key, right? So based on the current stance of the standard and the industry and the regulators, the major portion of the evaluation of biological safety depends on what you know about your device, right? The materials you're using, the processing you're using. You have to put that into a biological evaluation plan or an initial risk assessment where it takes into consideration all the available information that you have and defines what are the additional biological endpoints based on the duration of contact and the type of contact your device has, and then defines the supplementary information that you need to add into the available knowledge, right? Then you take that plan, you find your lab based on the expertise that they have. We really, as Matt said, just really do your homework, find the best lab that you can based on your needs, right? Needs for expertise, turnaround time, cost, whatever your need is. And then we really highly recommend that after all of the testing is done, you should have like a little summary report. We call it a biological evaluation report that basically takes into account the BEP. It talks about a little bit about what is the background information, what testing was recommended and why. Then it takes into account the testing that you did do based on the BEP, and then ties it up in a nice red bow that you can present to the regulatory body during your submissions.
Okay, so you start by making a plan that minimizes the number of tests that you need to do, leveraging existing information. Then you carry out whatever additional evaluations that need to take place. It might be testing, it might be other expert evaluations. And then you make a biological evaluation report that concludes all of this information in one nice package, says, this is the device, here are the risks that we found. We address those risks with these tests and expert evaluations, and therefore everything is biocompatible and we're good to go.
Yes, exactly. I'm going to high five you now.
Okay.
That was an amazing crash course you just put me and our listeners through. And I want to thank you so much for taking the time out of your day and providing this crash course into the preclinical and biocompatibility world. Also have to thank our listeners for tuning into this episode of RCA Radio. Be sure to subscribe to be the first to know when we upload the next episode of RCA Radio. Thanks again and have a great day.